Is this on the exam?
I was just kidding about that. I'm not that crazy.
That's what I was thinking.
I'm not that nuts, really.
For the equation sheet,
You know what?
the equation sheet for physiology, we don't have the equation for the both flow through the capillaries, like for the Sterling.
Correct.
Is that a hint that we don't need it because it was on the sample test? We probably need it. Okay, thank you.
I don't remember the top of my head, to be honest with you. I wish someone had pointed that out to me before.
For the exam, should we still know how to, like the details for the rearrangement, just like in the first exam for gene rearrangement?
Should we have nobody?
Should we know like gene rearrangement?
That was covered in the mail.
Okay.
Oh, you mean for like the TCR?
Yeah.
No, we already covered that.
Because we already covered it. Okay.
I didn't ask any questions about that. Once is enough. Once is enough. Yeah, Griffin, I'm at 3 o'clock. I'm at 3 o'clock, not 2 o'clock.
No worries.
I'm a little, my mind's a little,
I respond fast.
I have so many review sessions, so many office hours, and so many exams. Well, you're in both classes.
I respond fast, emails, no problem.
Another beautiful day in New England. Remember I told you there's only 10 nice days in the year.
They won't pass.
They won't pass, right? It's too late.
We looked out the window in our Spanish.
It's raining for 2 weeks. Except one day last week was nice.
It's true. And I had to work.
All right, a few announcements to make. First of all, I really don't care how you put your phones up here. I was only kidding the other day. I'm not that insane. But I see most people put them perfectly parallel to each other. So there you go. Number two, I hope all of you voted or will have voted. So make sure you do your citizenship obligation. And number three, there was something number three. What was it? I guess it wasn't that important. I remember I have office hours after this class, 4:30 to 6, but then at 6 to 7:30 I have review session for my physiology class, so I won't be able to meet with you 1 minute later than 6 o'clock. All right. So now we're going to do generation of memory T cells, which if you looked over some of the information gets kind of complex, and but it's actually kind of fun. And then we're going to start B cells and how they're educated to produce antibodies, which is a fascinating topic, really is still emerging. And I think this year's lecture is going to be more straightforward than other years, but we've learned some things in the last year which have clarified the area. All right. So there's a few cartoon experiments I want to show you. We're going to have some fun with them, I think. And I want you to look at this particular one, in which we have a transgenic mouse, and the transgenic mouse is only responsive to one particular antigen and restriction, as we know. So we inoculate our transgenic mouse with the same virus we've used a number of times. And when we do that, we then do flow cytometry, and all our T cells are going to be LCMV specific. We know that because it's transgenic, or virtually all of them will be. However, in flow cytometry, we're going to look at some cells which have a receptor for interleukin-7, high amounts of it, and then we're going to separate those from the same specific cells, but which for whatever reason express the interleukin-7 receptor in lower amounts, and we're going to differentiate them between low and high. We're then going to take the high ones and infuse them into another mouse, and then we're going to take the low ones and fuse them to a naive mouse. And then what we're going to see after 14 days or so, that the mice, which were IL-7 high, got the IL-7 high, had a lot more CD8 T cells, which were LCMV specific, than the ones which had low interleukin-7 receptor identification. What does that mean? That means it's really hard to generate cytotoxic memory T cells which do not have the receptor for interleukin-7. So interleukin-7 is evidently one means by which we can take naive cells and get their descendant cells to act as memory cells. And if a specific T cell, CD8 cell, which is LCMV specific for whatever reason, cannot respond to interleukin-7, it probably means it's a short-lived effector cell, which doesn't leave any descendants, and therefore doesn't have a long impact or long-term impact on the immune response. So to generate memory CD8 cells and probably CD4 cells as well, you probably need these cells, once they're exposed to antigen, to express interleukin-7 receptor. Now, the ones which are low can carry out the primary effector response. That's fine, but they don't leave any descendants, so you won't have any memory. So you really do need cells which have ability to respond to interleukin-7. Now, to find in an animal which is not transgenic, and an animal which is non-transgenic, a particular T cell, a particular restriction, and particular specificity was more or less something which couldn't be realized. People couldn't figure out how to find specific T cells in individual, not in culture, but in individual animals, or even humans for that matter, which have a certain restriction and certain specificity. Until they invented the following technique. And this following technique has now revolutionized the way you can identify cells from zillions of different cells in a particular organism, and that cell has a particular restriction and particular specificity. This technique was first used to get CD8 cells, and then it took a little bit of jury rigging to get it to actually work for CD4 cells as well. So most of the data that we have for this particular technique are for CD8 cells, but CD4 cells as well now are being fished out by this particular technique called the tetramer technique. And let's review what the tetramer technique is. The tetramer technique, first of all, we have a class 1 molecule that's been purified. So that's our specificity, our restriction. And then to that particular class 1 molecule, we add the peptide of interest for the T cell receptor we're fishing for. So we created, if you will, in vitro, the right restriction and the right specificity. Well, that's not easy to do, to get the class 1 molecule, or class 2 if you're looking for CD4 cells, and get the right molecule stuck in there, but biochemists have figured out how to do that. And then what they do as well, we add a molecule called biotin. Biotin can bind to another molecule with high avidity And the molecule that biotin, which is a vitamin actually, can bind to a molecule of high avidity is called avidin. And one of these molecules of avidin is actually found from streptococcus, so streptavidin. Let's get there in a minute. So we've taken our class 1 molecule, here's the beta-2 microglobulin. We've added the peptide to the binding clefts. Now again, we have the specificity and the restriction of the TCR that we want to find. And then we add biotin to it, and that's called biotinylation. So you add biochemically attached to this particular molecule, biotin, that's called biotinylation. You then expose this biotinylated class 1 molecule plus peptide to fluorescently labeled avidin or streptavidin. So this particular molecule, streptavidin, is fluorescently labeled. And what happens is this biotinylated class 1 plus processed peptide then will bind with high avidity to this fluorescently labeled streptavidin. And it turns out avidin has a valence of 4. So as it comes out conveniently, streptavidin can bind 4 class 1 molecules with the processed peptide. And for our story, all 4 class 1 molecules are identical, and all 4 processed peptides are identical to each other in our tetramer. Hence the word tetramer. And then what we do is we mix this tetramer with mixed cells in a population. Now, if you know how to do flow cytometry, we can gate our cells and we can pre-purify for lymphocytes which are either CD8 positive or CD8 or CD4 positive. That's pre-gated or gated. And let's say we gated beforehand for only CD8 cells. So now you have these whole mass of CD8 cells with a wide range of restrictions and specificities. So now we're going to use this tetramer to pick out from this wide range of CD8 cells with different restrictions and different specificities a particular CD8 cell which has the restriction and specificity we're looking for, i.e., a T cell with the T cell receptor which can bind to this particular restriction, this particular class 1 molecule, and this particular processed peptide. And then because this class 1 molecule and processed peptide appear more than once on the streptavidin, we can bind more than once to the cell. This can fold over so you can bind 4 times, so you can get more fluorescence. And then you put this through a flow cytometry machine, and then you can separate the cells which are fluorescently positive for this particular tetramer. And when you do that, you can, in essence, purify a wide heterogeneous population of CD8 cells. So now you have a single set of CD8 cells which all have the same restriction and all have the same specificity. So we fished out of a needle, we found a needle in the haystack. We fished out of these, again, this very, very heterogeneous population of CD8 cells, cells which have the exact restriction and the exact specificity that we're looking for. This is a beautiful, highly used, highly useful, very, very, very powerful technique. Okay? Used a great deal in modern immunology. So I urge you to understand what is actually involved. And again, this was first created to find CD8 cells, and with some modifications, now is also used to fish out CD4 cells, which has turned out to be more difficult to do them, but that can be done now as well. So it's a great technique. So now we're going to use our tetramer technique in the next cartoon experiment. So let's use this tetramer technique again in the next cartoon experiment. I want you to read this, what's going on, and I want you to figure out what's happening, and I want you to figure out the technique of the tetramers, how we actually picked out these cells. So why don't you read this cartoon and figure out again how the technique was employed in this situation. Again, the topic we're discussing here are memory T cells. Keep that in mind. And by the way, LM is Listeria monocytogenes, a nasty bacterial infection. I want you to figure out how they got from panel 1 to panel 2 using the tetramer. And ditto for from 2 to number 3. You can talk louder if you want to. As long as you're not talking about the weekend. It's only Tuesday. It's going to rain anyway this weekend, for sure. Oh, the third thing I wanted to ask you, did everybody get your flu shot? Everybody get your flu shot? Because it turns out it's a good match this year with the vaccine and the strains going around. So get your flu shot. I don't want you to give it to me. All right, class. All right, so we have a wild type mouse and a class 2 negative mouse. You should know this pretty well for the exam on Thursday, even though, remember, today's lecture is not on the exam, but nonetheless, you should know what that means. So if you're class 2 negative, will you have any CD4 cells? No. Okay, knowing by negative and positive selection, you won't have any CD4 cells. So then, after 7 days, what happens, class? Tell me, someone raise your hand, what happens in panel number 2? Someone tell me, yes.
The mice make CD8 cytotoxic T cells.
How did you find that out? How did you get those cells? How did you know that? How did you get to panel 2? How did you get those cells to prove that point?
How did I know that, or how did the...
Well, how did they do it?
Using the tetramer.
Right. So explain that again. So what did they do for the tetramer? What did they do?
So they took the peptide that they were looking for.
Which is what?
What?
Which is OVA.
Yeah, which is OVA. And they connected it with a class 1 MHC.
Right.
And biotin, and then the biotin linked to the streptavidin and made a tetra for identical class 1 MHCs and 4
Right.
identical peptides.
Right.
And that and they injected that.
So they did flow cytometry because you
Yeah.
have the fluorescently labeled streptavidin.
And they looked
right?
to see which cells.
So they could snag out these cells, right? And then they can count them, right? So they can count them. So we can get counts by doing flow cytometry, and by doing the tetramer technique, we can get the exact cells with the right restriction and right specificity and prove that they're actually there. So in both mice, true or false, we had OVA-specific CD8 T cells, whether or not they were CD4 positive or negative. Yes. We could do that in mice, at least. Whether this would work in humans is probably not the case. But then we move to the third panel, and what happens in the third panel? This is important. We use the tetramer because now we get number of cells. In the third panel, we're looking for memory. And if you didn't have CD4 help originally, do you generate memory CD8 cells? No, we do our tetramer technique once again, and cells which do not receive class 2 help via a CD4 cell were failed to generate memory cells, and therefore their numbers of OVA-specific CD8 cells were woefully low. Yes.
How can you distinguish a memory cell from a...
There are. We're going to learn that in about 10 minutes.
Okay, so
There are markers. Yes,
Okay.
there are markers. Many markers. We're only going to learn a few of them. But there are markers. Thank you for asking. The question is, how do we detect memory cells? And we're going to learn some markers to identify memory cells. Not all of them. There's many of them. And we're going to learn a few. Okay, good question. Other questions? All right, so now you know a new technique, another technique, tetramer technique. All right, well, here we go, the answer to your question. So how can we determine very simplistically a naive cell from the effector cell to a memory cell? And you look for various CDs on their surface, and we're not going to worry about CD44 to any great extent. The two molecules of interest to us for today's discussion are CD62L and CCR7. CCR7 is a receptor for a chemokine. And cells which express CCR7 respond to a particular chemokine which draws them to wherever that chemokine is being produced. And the chemokine which attaches itself to CCR7, which is usually expressed in pretty nice concentrations in lymph nodes. So a cell which expresses CCR7, its home away from home will be a lymph node. On the other hand, CD62L is a molecule which sticks. Okay? This molecule CD62L has another name. Its other name is called L-selectin. And L-selectin is a lectin. And if you remember, the definition of a lectin is a protein which can bind to a sugar. So that's just a lectin, all right? So lectin is a protein which can bind to a sugar. The only thing I'm leaving out there is a lipid. I got sugars and proteins. Anyway, CD62L can bind to, is a protein which can bind to a sugar. And it turns out, if you remember this little bit of histology, blood vessels which go to lymph nodes have capillaries. And after the capillaries, it's called a post-capillary venule. And post-capillary venules have plump endothelial cells instead of flat ones. And because they're plump, they're called high. Not for chemically induced high, but because they're plump. That's called high endothelial venule. HEV, not HIV. HEV, high endothelial venule. And high endothelial venules have a sugar on their surface that L-selectin can bind to. So CD62L on the lymphocyte will bind to a sugar which is present, a glycosylated protein, on a high endothelial venule. Okay, so ignoring CD44, we have CD62L, which is CCR7. Okay, effector T cells, which are in the periphery, do not want to home in to the lymph node, so they don't have much CD62L. They don't want to go to the lymph node. They want to stay and carry out their response in the periphery. So they have low CD62L. They don't want to go to the lymph node again. They don't want to respond to the chemokine produced in the lymph node. So they have low receptors for the chemokine which binds to CCR7. So they stay in the periphery. Effector memory cells want to stay in the periphery, so they don't have these two molecules expressed in very high amounts. They stay in the periphery. And central memory means they want to stay in the lymph node primarily, so therefore they have both molecules in high amounts, CD62L which is the lectin or selectin, L-selectin, which will bind to a sugar on the high endothelial venial and stay in the lymph node, and CCR7, which is the receptor for the chemokine expressed in high amounts in the lymph node. So that works nice. So there's an answer to your question about what memory cells look like and naive cells, but it's not that straightforward. Nothing in immunology is. So let's look at this next slide. And I want you to figure this one out. I'll give you all the clues, and then we're going to make this even more complicated on the next two slides. But for the moment, let's step up our story, make it a little more complicated. And we're going to do two different flow cytometries, one for CD4 cells, one for CD8 cells. We're going to use CD62L and CD45RA. And I told you what that is down here. I typed it in, so you can read that, please, and self-educate. An autodidact is a person who teaches themselves. Remember, for the rest of your lives, you're going to be autodidacts, self-taught. After you spend $280,000 here, you'll have no other choice. All right, at any rate, set this up, please. For the rest of your life, you're going to read journal articles, books. And figure everything else out in life on your own. I'm trying to make you do that now. We already talked about CD45, remember? Okay, let's someone volunteer. Let's do our CD4 panel. And I gave you the answers here, but I want someone to tell me anyway what it actually means. So in the upper right, we have our naive cells. Why are these cells naive in the upper right? That means they express lots of CD45RA and they have CD62L. Why do you think the naive cells are in the upper right? The high expression of both these markers. Yes.
Is it because they need to be in the lymph nodes so that they can get specific?
Perfect answer. Exactly right. They need to be in the lymph node, so they need the CD62L to get into the lymph node, right? And since they're naive, they're not memory cells, so the isoform of this phosphatase they're going to have is exactly right, CD45RA. So they're going to have lots of CD45RA. All right? Who wants to take a shot at the upper left? Using the same bit of analysis. Yes?
They don't have the CD45 anymore because they aren't naive anymore, but they still have the CD62 because they still want to be in the lymph.
Right, so they still want to go to the lymph node because they're central memory. And instead of having CD45RA, the isoform of CD45 they have is CD45RO. And CD45RO is not being picked up by our flow cytometry because our antibody doesn't bind to it. And CD45RO actually has fewer exons used than CD45RA. It chops out some exons. It's a shorter molecule, but it's actually more powerful. It's a more powerful phosphatase, so it can be activated more quickly, the signaling. So very good. So in the upper left, we still want to go to the lymph node because we're central memory, but now we have the imprimatur of a memory cell. So instead of being RA positive, it's RO positive, but again, our antibody doesn't pick that up very well. So that's why you're in the upper left. All right. What about the lower right? Lower right, yes.
They're negative for CD62, so they don't want to be in the lymph nodes, they want to be in the periphery
Right.
to be able to be effectors.
Right, and there's still probably effector cells short-lived and not a memory cell, so they're therefore the CD45RA high. And lower left are lower left are what? Someone, lower left are what? Effector memory, because they're effector cells, but they're memory type of effector cells because they don't have the RA isoform, they have the RO isoform, and our antibody doesn't pick that up very well, so it dumps those cells in the lower left. So we can distinguish 4 different cell types by using these 2 different antibodies and then doing flow cytometry, either pre-gated for CD4 positive cells or pre-gated for CD8 positive cells. We get more or less the same phenotypes. Now, however, this is old stuff. You can see this is from 10 years ago. This isn't the state of the art of the situation. Now we're going to complicate it. All right, so I want you to read through this particular figure because there's so many things on this particular figure. I can go through it very quickly, but I want you to enjoy it and assimilate it into your engrams in your brains. So look what's going on, all the different cell types here. We're going to have this many cell types. 4. Central memory, effector memory, resident memory, and these are 2 new types we haven't talked about yet. Notice what's not here. Naive cells are not listed here, nor are effector cells. So we actually have 2 new phenotypes. 2 new phenotypes are resident memory and peripheral memory, and I'll learn you what they all do in a minute. But I want you to look at what distinguishes one from the other. So go ahead and look at it. Seen this before. This is a new chemokine receptor. You haven't heard about that one. CD103 is an adhesion molecule, and CD69 will keep a particular lymphocyte in a particular area and not leave. CD69 will keep a particular lymphocyte in a particular area and not leave. Let me just say that to simplify it. Adhesion molecule, CD103, CD69, it keeps the lymphocyte in situ, keeps it where it is, in residence. All right, everybody look at this complex figure. Let's go through it together. I'm going to help you on this one. And what happens is, here's a we're not going to worry about naive cells here. We're not going to worry about how we're going to activate those naive cells. We're going to learn that either today or after your exam next Tuesday. But we have pre-central memory cells, and these pre-central memory cells, people think do not express 62L, but the ones which actually are central memory express CD62L. And CD62L cells, preferentially if they're memory, go to lymph nodes, go to the efferent lymphatic, this you should know anyway from the first exam, then go into the thoracic duct, then back to the blood, and then home in again back to the lymph node. So central memory preferential tour of our bodies is mostly staying in the lymph node tissue or lymph tissue. But recirculating through the lymph tissue, the blood, and back to the lymph tissue. Okay? Then you have the CD62, another set, which are peripheral memory cells, and the peripheral memory cells which express CD62L are really essentially the same as central memory. So we will just say these 2 cells are essentially the same. You can see, again, their map. But the true peripheral memory cells aren't the CD62L positive. They're essentially the same as central memory. just to complicate the terminology, unfortunately. The ones which are true peripheral memory, however, are CD62L negative. They don't want to be in the lymph node too long. So where they go is they go through the peripheral tissue, looking for things to interact with, get swept up into the afferent lymph, bypass the lymph node, don't hang around there, boop, go back to thoracic duct, the blood, and then go back to the periphery. Okay? And these cells have this particular chemokine receptor. That's how we can distinguish. Again, answering your question, that's how we can tell these are memory cells. They have this particular chemokine receptor. And finally, no, two more. Another one are called resident memory cells. Resident memory cells have this particular phenotype, CD69 positive, which allows them to stay in place, to not escape where they are. It forces them to stay where they are, along with CD103 positive, CD3103, which again is an adhesion molecule. It keeps them in place. And the resident memory cells are interesting. They were probably seeded early in life. And they were lymphocytes which responded to a particular antigen and just hung around forevermore at that particular site where they were first activated. So they can pounce on that microbe should it appear again. And they're self-renewing. So they undergo replicative phases and therefore never die out. So these cells are self-sustaining. So they're there for the rest of your life. And finally, the last one is the effector memory cells. And the effector memory cell seems to spend all of its time in the blood and the spleen, looking for intravascular and splenic microbial invasion. So we have, again, these four phenotypes. Central memory, hang around in the lymph nodes. Peripheral memory, in the periphery, in your skin, in your intestinal tract, respiratory tract. Effector memory, mostly in the blood and spleen. And resident memory, hang out in the tissue for the rest of their existence. And they're self-renewing. Yes? So every part of your body is now protected by a particular type of lymphocyte, which is a memory lymphocyte, which can respond to an antigen very, very quickly. Ilana?
The central memory and peripheral memory, after it goes through the thoracic duct, that into the blood and the spleen, does it ever go back into the lymph nodes or the peripheral tissue?
Well, the central memory certainly goes back into the lymph tissue, yes, because it has CD62L positive. The PMs, which are CD62L negative, just bypass the lymph node and go back to the periphery.
Oh, but they do go back.
Yeah, they have to do that, but they don't spend any time in the lymph node. They just go right past through it. Okay. Now, I want you to read the following figure, though, just to make sure you understand what all these cells do. And we already went through these, and resident memory not listed here. There's also RM, which is talked about down here. So there's four of them: central memory, effector memory, peripheral memory, and resident memory. So all four we just talked about. Now, important for you to talk about, to read is this sentence. I want you to read this sentence and think about it. There's a lot of information in this very short sentence, which I'll tell you about, but I want you to think about that, what that means. It has very important ramifications, which we're going to come back to later in the course. And RMs are just what I exactly said already. And these CM and EM and PMs are written out for you what they do. I already told you what these do, and I already told you what RMs do. This is the new bit of information. It's a good slide because, again, it reviews what everything I just said. All right, let me ask you a question, going back to the sentence. in the intestinal tract, what's the favorite isotype of antibody? What's the favorite isotype at the intestinal tract? Ig what? A. And by the way, for B cells to undergo class switching, is there need T cell help to undergo class switching? Do B cells need T cell help to undergo class switching? The answer is a one-syllable word with 3 letters in it. Yes. And it turns out if you have a T cell, which is going to help a B cell become, let's say, IgA positive, you want that T cell, once it's a memory cell, to always circulate back to the intestinal tract, because that's where you want the IgA to be produced. So if a cell first encounters antigen in a lymphatic tissue at a particular site in your body, it's then given a series of molecules on its surface, which will always cause it to circulate back and enter that particular tissue. So if a T cell first became educated, not positive, negative like the thymus, but first became activated by an antigen in your intestinal tract, and it circulates, it will always return to the intestinal tract. If that T cell first became activated by an antigen in the respiratory tract, it gets certain addressed molecules on its surface, which will always return to the respiratory tract. If you have a T cell which is first encountering antigen in your skin, it will get certain molecules placed on its surface that will return only to the skin, not to the intestinal tract, not to the respiratory tract, et cetera, et cetera. So the immune system is very efficient. Once a lymphocyte encounters antigen for the first time, it acquires a set of surface molecules that in the memory state will preferentially take that memory T cell back to that tissue where it first encountered the antigen. So does that make sense evolutionarily speaking? Yes, because if I encounter an antigen in my gut, and I'm a memory cell, more than likely that same microbe will invade my gut, not my skin, and vice versa. So microbes have preferential places they want to go in your body. So therefore it makes sense that if you encounter antigen in one place in your body, you should have an address to send you back there as a memory cell, so that you're more likely to encounter the antigen to which you already are sensitized to. Everybody get that? So it's a really nice evolutionary system. Okay, yeah, question.
Do you get, for example, for a specific bacterium, do you get all 4 types of memory cells for the specific?
That's a very good question. And it depends. For example, if that microbe is able to set up housekeeping everywhere in your body, in your spleen, in your blood, in your lymph node, in your peripheral tissue, then I'd say yes. But if it's preferentially going to go just the peripheral tissue, it will probably be a peripheral memory cell or a resident memory cell, which would be there. So it depends.
So is the purpose of circulating back just to engage or induce class switching for more B cells? Or?
No, it's just to get that B cell, maybe not do class switching, but it's already IgA, you want it to stay as IgA, but then you want to, it's in a resting state, you want to activate it again so that it can start making new plasma cells, which make new IgA, which then can help you fight off that particular microbe. So for a B cell to get reactivated, it needs T cell help.
Oh, so if the antigen is presented another time, the recirculation.
Right, you need, again, that T cell help. So therefore you wanted to go back to the gut, the T cell, so it can help the B cells of that site resynthesize IgA.
Oh,
okay. So yeah, it's quite a nice system. And therefore I do want you to know. And then there are cells which stay there forever. They never leave. The resident memory cells, which really can act very quickly to that antigen and get you a foothold in fighting off that particular microbe. So the RMs are very, very important as well. Okay, so the 4 cells we're worried about are central memory, effector memory, peripheral memory, and resident memory. And I'm ignoring naive and the short-term effector cells. Okay? All right. Let's go to the next topic. Remember, these particular bits of information will not be on your second test on Thursday. Memory T cells, that will be on your final. And now we're going to have B cell development. All right. Now, just like everything in immunology, the 9 types of lymphocytes and types of subdivisions of cells we have is highly, highly complex. Turns out there's more than one type of B cells. The 2 main divisions of B cells, not the only types of B cells, are B1 and B2. B1 T cells apparently make a bridge between the innate and adaptive immune response. They make antibodies which have broad specificity instead of very restricted, and therefore can bind to a wide range of microbes, but have a much lower affinity or avidity. So they can work in the short run, but not in the long run. So B1 cells, some consider really part of the innate immune response, or others consider it a bridge between the innate and adaptive. We're not going to talk about B1 cells at all in this course, other than what I just said. So don't ask, we're never talking about them again. Well, you could ask, but we're never talking about them again. So the other ones that we are going to talk about are B2 cells. And the B2 cells are the classic ones which we've been describing, or I've been describing throughout the semester. That's the ones we're going to be talking about today. Okay. All right, and here's somehow how B cells develop in the bone marrow. If B cells are induced to express Notch, stem cells are induced to express Notch, they'll turn into T cells. However, if you downregulate Notch in your stem cell, you'll end up with a B cell lymphocyte instead of a T cell lymphocyte. We've already talked about Notch before. In any case, you can see that the various molecules are needed to help us make a B cell. IL-7 is certainly involved. So no more needs to be said about this particular slide. No need to memorize it other than IL-7 is important in helping us get a B cell development at this stage. And you don't want Notch. All right, but this slide is a review of what we've done before, so I want you to quickly review it. And we're going to go through it pretty quickly because all this information we've had before. All right, you reviewed it enough. All right, let me ask you quickly, what goes first, heavy chain rearrangement or light chain rearrangement? Heavy, great. Okay, so then we complete the heavy chain rearrangement, then we express the mu heavy chain with surrogate light chain, remember that? And then this complex causes the B cell to proliferate, and then when it proliferates, we then do the light, after it stops the proliferation, we then do light chain rearrangement. And by the way, when is TDT operational? During heavy chain rearrangement or light chain rearrangement? Quick review. When is TDT expressed? During heavy chain rearrangement or light chain rearrangement? Heavy, right. Can we actually get, however, N nucleotide addition in the light chain as well? Yes, by a different mechanism, probably a polymerase, a DNA polymerase. So, good. Okay, mu polymerase. All right. Now we have light chain rearrangement. Okay, now we have the IgM expressed on the surface. M stands for membrane bound, IgM. And then at this stage, you can respond to a self-antigen. And if you only have M-IgM on the surface, you're naive. And you're exposed to a self-antigen at this point. The B cell will kill itself. So it's at the single positive IgM phase when the B cell can undergo apoptosis if exposed to a self-antigen. Let's leave it at that. Once, though, you change the RNA processing, so you not only have the isotype IgM, but you also have the isotype IgG both expressed. We're now in the mature B cell, and that is when we leave the bone marrow. Let me ask you a question. When you have the IgM and IgG expressed at the same time, do they have the same idiotype? If IgM and IgG expressed at the same time, do they have the same idiotype?
Yeah.
Yes. Single B cell will only have one idiotype, normally. You may have two different isotypes, but it'll always have one idiotype. All right. Then they get antigen stimulation in the lymph node or another lymphatic tissue, and then you get IgM first, and then you do class switching to the various the other isotypes. Okay, that's the life of the B cell. And then, of course, it's not as simple as that. Once a B cell leaves the bone marrow, we have two subdivisions of these B cells, transition cell 1 and transition cell 2. And transition cell 1 have low amounts of IgG. They enter the spleen. They leave the spleen as T2 cells. And as T2 cells, they then go to lymph node to get their final development. And then they finally have high amounts of IgD and are mature B2 cells. All right. Whew. Now these next 4 slides, I love these next 4 slides. 1, 2, 3, 4. And what they did in the next 4 slides is they used 2 different techniques. They used flow cytometry, and they did genotyping, sequencing of DNA. They did 2 different things, flow cytometry first, and then the cells, which were in a particular quadrant, they then did nucleotide sequencing, or gene sequencing, to figure something out. Now I want you to figure out this particular graph, or figure, based on the information I've typed in here. And I want you to figure out this particular figure but based on the information I've typed in here. These are really fascinating slides, research done by individuals who did a lot of hard work. So I'm going to look at this one, and this is the cartoon depiction of it. This is the real data, and this is the cartoon depiction of it. So I'm going to look at them, look at the flow cytometry, and if you can't figure it out by the flow cytometry, what I've typed in, then look at the cartoon flow chart. Okay, so go ahead and do that right now, and I want to hear a lot of talking because these are fun experiments to interpret. And remember, if you want to go through the 4 slides, you can do it on your own. If you have them in front of you. Remember, my goal is to turn you into real immunologists, not just note takers. Hope you all do that. By the way, if anyone can figure out how to get the slide so it's not cut off at the top, I'll give you 2 extra points in the course. I can't bribe you with money, I'd lose my job, but I can give you 2 extra points if you figure out how to not get that part fucked up. By the way, in this section, we're going to have a lot of experiments on what you to interpret rather than me. So,
Right, but the next
Oh, they're mature, yeah. Oh, so that makes sense that we have.
By the way, next Tuesday is a Tufts Monday, right?
No.
I was just messing with your minds, I know that. Yeah, it's Tuesday. However, there's no class on Monday for any Monday classes you have, because it's Veterans Day. And remember, on Sunday is the 100th anniversary of the end of World War I. And by the way, the last person to die in World War I died at 10:59 a.m. European time on November 11th, 1918, 1 minute before the war ended. And just think about how horrible that is. I hope you read some of the reminiscences, histories of World War I, a war that should never have been fought. I'm going to teach you history here. At any rate. Millions of people died for no reason.
Or the CD14 negative.
Yeah, it would be like it would be like
Oh, so they're all negative because these are very
All right. HSA, horse serum albumin. Is that what HSA stands for? Horse serum albumin. Yeast stable antigen. Okay. BP, British Petroleum. No, it's an aminopeptidase. It means it chops up proteins at the amine, starting at the amino end. All right. So we have 4 flow cytometry centers here. One to the left, one to the right. Stand up, sit down, fight, fight, fight. Okay. One in the upper right to the left, and one to the upper right, further to the right. All right, so now we have A, B, C, C prime. Boop, boop, boop. So they took the cells from here in class. What did they do with the cells here? They did analysis, gene analysis, and what did they find out as far as VGJ joining? No gene arrangement. So these must be the earliest what? Earliest what class? B cells. Oh, then they found out in group B, okay, which has this particular phenotype of high HSA and low BP, they had DGA rearrangement. Remember I told you DJ rearrangement takes place before VD rearrangement. And then finally C is VDJ rearrangement, so they're high for both BP1 and HSA. And then they shift to even a higher phenotype of HSA and the same amount of BP1. At this point, we have the mu chain being expressed and the surrogate light chain being expressed. Well, obviously, you wouldn't use these 2 markers for doing this flow cytometry because these things aren't expressed until you get to C prime. So there wouldn't be anything you would find. So you have to find some other markers which would identify these early B cells, and again, that's with HSA and BP1. And then they did pre-gating, by the way, with CD43. CD43 is just an adhesion molecule, okay? And B220 is a glycosylated form of CD45. So B220 is yet another isoform of this phosphatase CD45, but it's only found in B cells. So this particular glycosylated state of CD45 is only found on B cells, similar to the one found on T cells, but has a different glycosylation pattern, and therefore it's given a different name of B220. Okay? It's similar to the phosphatase found on T cells, but again, has a phenotype which is slightly different, an isoform which is different. All right, and again, all this information is repeated on the cartoon. All right, now look at this one. We're looking at different markers now. You're looking at IgM and now again B220. B220 stays more or less the same for this end of our story, but IgM levels change. Remember, we already had M being expressed on the previous slide. We had M being expressed, but not IgM yet. Remember, these experiments do both flow cytometry and genetic analysis, nucleotide sequencing, gene sequencing, whatever term you want to use for it.
Where's CD43 on
Sorry, what?
CD43?
Remind us, that means we pre-gated them, and then when they lose CD43, we know they're a more mature type of B cell. So,
So you
compared to the
disperse.
previous one, right? Yeah. So they still have 220, but they no longer have 43. Okay, I don't hear much conversation here, so I guess we figure it out. So what goes on in our story here with D, E, and F? In D, very little IgM. Okay, what happens at this stage? We have the heavy chain being expressed with the surrogate light chain, but VJ joining begins. In E, what happens in E? We had IgM now, because now we have both the heavy and light chain. For the first time now, we can say we have IgM being expressed at this point. And then explain the shift back to the left with F. Hypothesize. It's not stated on the cartoon. What's your hypothesis? Why did we get a shift which looks insignificant, but it actually was a significant shift to the left? So what was that shift to the left? Where do you think that came about from? The same two people, other people? Somebody? Yes.
So IgM is produced first, but there's a class switching to IgD.
Yes?
So
Not class switching.
Sorry,
That's not the exact. In this case, we don't switch to IgD. It's the only time we don't. Now we co-express because it's on that same chunk of DNA. There's no switch site in front of IgD, so they're co-expressed, co-transcribed, and then eventually co-translated. So it's not true isotype switching.
so there's probably going to be less IgM produced if you're producing both IgM.
Exactly right. Perfect. Exact correct answer. So what happens is, in this case, the only isotype you have is IgM, and therefore that dominates the transcriptional and translational machinery. But then when we co-express both IgM and IgD, there's just less IgM available because we're spending time expressing IgD, so we get a shift to the left for IgM. That's exactly correct. Perfect analysis. And again, somewhat of this is explained on the next cartoon. All right. Now I want you to figure this one out. I've given you all the information here, except let me just say DNP is dinitrophenol. Dinitrophenol. Okay, dinitrophenol. How many people took organic chemistry in this class? How many people took organic? Keep your hands up. Keep them up. Tell me how to draw a dinitrophenol. Make me a phenol. How do I make a phenol? What am I going to do, class? And then dinitro. That means what? Actually, this. Can I put another one on? All right, and what do I abbreviate dinitrophenol as?
DNP.
Okay, figure it out the rest. And by the way, oops. What's this? Or, OP, ortho, para, meta, OPM. Am I wrong? Ortho, OMP, you're right. OMP, sorry. Oops. All right, OMP. All right, OMP. Oh, better. Sorry. Ortho, meta, para. Okay. Can antibodies distinguish between ortho, meta, and para? Yes, they can. Okay, so keep that in mind. We can distinguish between ortho, meta, and para. All right. Okay, does anyone know what this is? Anyone know what this organic molecule is? This is the ether bunny. All right. Does anyone know what that is?
MD? No.
Orthodox. This is orthodox. Get it? Anyway, have fun with organic chemistry. All right. By the way, does anyone know what organisms which are in archaea? Does anyone know what their phospholipids in the membrane are? Ether linkages or ester linkages? Does anyone know that? In archaea? Ether linkages. So these old organisms have ether linkages instead of ester linkages. Kind of interesting. All right. Anyway, getting back to immunology, we figured this out. I was talking the whole time, but what is this? Well, we take our mouse in the middle. We inject it with DNP-BSA, right? Which one's the bigger molecule, DNP or BSA? BSA, what does that stand for? Not Boy Scouts of America. What does it stand for? Bovine serum albumin, okay? And then the mouse on the top there, little a to the left. We give it secondary DNP-BSA. We get a secondary anti-DNP response, okay? No shock there. Now we distinguish the primary from the secondary antibody response. I typed that in for you. All right. What happens if we give DNP OVA though to the mouse? The mouse has seen DNP before, but it has not seen the OVA before. What type of antibody response do we get? Primary or secondary response? Primary. So why is that the case? The animal's seen DNP before, so why do we get a primary response instead of a secondary response? Think about that for a minute. So we're going to lead up to figuring out how B cells actually can end up making antibody from these little bits of data. All right, now let's go from middle animal to C. And C, this mouse gets ovalbumin. And now this mouse gets DMP-OVA, and do we get a primary or secondary response? Secondary response. So that must mean to get a secondary response, not only must you have seen the hapten molecule, which is that small, very small organic molecule, which is difficult to make an antibody against, but you also must have that animal have seen the protein carrier that carries that hapten into the organism. Okay. Now look at this one. In this case, the X-rays, we lethally irradiate it, and now we are transfusing cells and reconstituting that animal and saving it from death by either the animal receiving NIP. NIP is this long, unpleasant pronouncing name. It's also a hapten. It's a very small molecule. You can see that it's a very small molecule, even though it's a long name. And figure this one out. There's a lot more to figure out in this one. Let's see if you can do that. There's some very central bits of information telling us how B cells know and learn memory against antigen. All right. This animal is getting NIP-OVA injection. We then take our naive mouse, we lethally irradiate it, we then transfuse spleen cells into its tail, into this naive mouse. We then give it NIP-OVA, and lo and behold, we get a secondary response because it has the memory cells from this first animal infused into it. That's no big problem to figure that one out. By the way, we'll assume this is an inbred strain of mouse, so we don't have to worry about haplotype differences between them. All right, experimenter 2. We lethally irradiate this second animal. We give it spleen cells from NIP-OVA exposed mice. We now expose this to NIP-BSA. We learned this on the previous slide. In order to get a secondary response against NIP, against the hapten, you have to have seen both the hapten and carrier molecule before. Does anyone remember what the carrier molecule means? I told you this before. You have these protein molecules, and what do we have stapled on the surface of these protein molecules? Many, many, many times. The hapten. So you have the hapten stapled chemically to the protein carrier, and therefore the immunodominant epitope becomes the hapten because it's repeated zillions of times on the surface of that large protein carrier. So that becomes the immunodominant epitope. It's repeated zillions of times on the surface of that protein carrier. Okay? All right. So that means not only must you have seen, though, the hapten before, you must also have seen the protein carrier before to get a secondary effect. Experiment number 3. We lethally irradiate this animal as well. We then take spleen cells from a BSA mouse, And then also give it cells, lymphocytes from an NIP-OVA mouse. And then give it NIP-BSA. And do we get a primary or secondary response? We get a secondary response. Because this animal, one way or another, by the transfusions, has cells which have seen both the hapten and carrier before. Maybe not at the same time, but the cell sources have seen the hapten and carrier before. And finally, in the fourth experiment, is something which is going to lead us to the next bit of information we have to worry about. Experiment number 4, the animal gets BSA, spleen cells, lymphocytes, which are responsive to BSA. We also give it spleen cells, lymphocytes, which are responsive to NIP-OVA. And now we immunize our animal with NIP, horse serum albumin, that's the carrier now, plus BSA, which it has seen before, and we get a primary response. Why? Because hapten and carrier, in order to give you a secondary response, not only have the cells in that organism's body must have seen previously both the hapten and the carrier, but in order to get a secondary response, the hapten and carrier must be physically linked. The reason is, let's see.